All right, gentlemen, today is Tuesday, April the 7th, and we are going to talk today about changes or mutations in chromosome number or structure that can cause genetic disorders. Okay. As you are well aware, that diseases, syndromes occur all the time in human beings, and many of these are because of chromosomal abnormalities that occur during meiosis or the crossing over process, and sometimes other times during the development of a fetus. And so that's the point of today's lesson 15.4. So if there are, we know that under normal circumstances, a fetus is formed or during fertilization when 23 chromosomes from the mother and 23 chromosomes from the father come together at that fertilization moment. However, if there is a major change in the number of chromosomes or even the structure of chromosomes, that typically will lead to miscarriages. And so the developing fetus will be naturally miscarried. In plants, it turns out that genetic changes like this are really not as dangerous or as lethal as in mammals or animals. Now, how do we get an abnormal number of chromosomes? Well, the most likely way for this to happen is during a process called nondisjunction. During meiosis, the homologous pairs of chromosomes should separate, all right, during meiosis 1 and then again during meiosis 2 to form the gametes. However, during nondisjunction, these pairs of chromosomes do not separate like they should. And as a result, one of the gametes will receive two of the same kind of chromosome, and another gamete will receive no copy of that chromosome. Now, this is an illustration of two different ways that nondisjunction can occur. On the left-hand side, the nondisjunction event occurs during meiosis 1. And so on one side, or one of the eggs ends up with 4 chromosomes, six chromosomes, and the other end ends up with only 2. The second kind of nondisjunction is shown here on the right-hand side, where the nondisjunction event occurs during meiosis 2. Now, this can lead to one set of normal eggs, or sperm, as shown here. And what is going on? I hate this mouse. I'm just going to leave my hands off the mouse. Okay, so if you look at the left-hand side where nondisjunction occurred during meiosis 1, you end up with a haploid plus 1 set of gametes on the left side and a haploid minus 1 on the right. In the case where nondisjunction occurs in meiosis 2, you end up with an N plus 1 and an N minus 1 on the left, and then 2 normal haploid gametes on the right-hand side. All right, and so nondisjunction can occur either during meiosis 1 or meiosis 2. Now, if there is a fertilization of gametes in which nondisjunction occurred, we call the condition that occurs aneuploidy. And essentially, offspring with this condition have an abnormal number of a particular chromosome. They can have an extra copy of a particular chromosome, or they can be missing a copy of a particular chromosome. If they're missing a copy, they are called monosomic zygotes. They have only one copy of a given chromosome. On the other hand, if they have 3 copies of a given chromosome, they are referred to as trisomic. Now, you can also have a condition where the entire set of chromosomes is non-disjoined. And so we call this polyploidy. Polyploidy is a condition in which an organism has more than 2 complete sets of chromosomes. If you've got 3 sets, it's called triploidy. All right, not trisomy, but triploidy. And tetraploidy is when you have 4 sets of chromosomes. Now, these polyploid conditions are commonly found in plants, but not in animals. They are typically lethal in animals. And it turns out that polyploids are more normal in appearance than aneuploids. Another thing that can happen, completely different from a non-disjunction event, is a breaking of a chromosome during the meiosis process. And this can lead to 4 different types of changes in chromosome structure. We'll start with the easiest one, which is a deletion of a particular chromosomal fragment. So you're missing a gene or maybe several genes. A duplication occurs when, for some reason, a segment of a chromosome is reproduced, and so there's 2 copies of that segment, or maybe even more than 2 copies of that segment on a given chromosome. An inversion occurs when the chromosome breaks in 2 places, the fragment flips over and then reforms into the chromosome. This can lead to genes that are essentially backwards and not read correctly in the production of proteins. And finally, translocation will take a segment off of one chromosome and essentially glue it onto another chromosome. This slide shows all of those 4 different types of issues. All right. In the first case, up in the upper left-hand corner, you've got a deletion. All right. And notice that gene D is removed during the process, and so it doesn't exist in the final product here. In part B, duplication, genes B and C are essentially duplicated, and so you end up with a chromosome that's got BC, BC, and so it's got extra copies of those genes. In situation C, we've got the sequence BCD, but if they're cut here and flipped over, we now have the sequence DCB, and then finally, in a translocation, notice we've got genes A and B on this chromosome, and M, N, and O on this chromosome, and they essentially switch places. So this one goes to a given chromosome, and this one goes to a different chromosome. Okay, and so this can lead to some serious issues, and we call them disorders or syndromes. Alterations of chromosome numbers or structures are associated with very serious disorders. Some types of aneuploidy appear to upset the genetic balance less than others, which can result in individuals that survive to birth and even beyond. However, most of them lead to lethality in the uterus, in other words, a miscarriage. However, surviving individuals will have a very specific set of syndrome symptoms, and we'll talk about some of them next. Probably the most well-known of all of these syndromes is trisomy 21, which is today called Down syndrome. This is an aneuploid condition where each individual has 3 copies of chromosome number 21. If you look at the karyotype here, here's chromosome number 21, but notice again there are 3 copies of it. It typically affects about 830 1 out of every 830 children born in the United States. And although the reason is unknown, the frequency of this syndrome tends to increase with the age of the mother. Aneuploidy can also occur with the X and the Y chromosomes, and so this can lead to a variety of different syndromes. The first is Klinefelter syndrome, which produces a male offspring, but he's got 2 copies of X chromosomes. There's also a scenario where 2 Y chromosomes can be found in males, and about 1 in 1,000 males exhibits this, but there are no symptoms of this for some reason, probably because the Y chromosome doesn't really carry many genes. We can also have trisomy X here. Again, 1 in about 1,000 females will be trisomy X. They are generally healthy with no unusual characteristics, but they do seem for some reason to be at risk for learning disabilities. Finally, monosomy X produces what we call an XO female. Again, no Y chromosome, so they're female. And these females are sterile, although there's not many other physical characteristics here. Now, disorders that can be caused by can also be caused by structurally altered chromosomes. A common structural change is a translocation between chromosome 9 and chromosome number 22. And this results, and you can see that here's a piece of chromosome number 22 on the end of chromosome number 9, and a piece of chromosome number 9 on the end of chromosome number 22. This is known as the Philadelphia syndrome or the Philadelphia chromosome. Cri du chat, or the cry of the cat, results from the deletion of specific genes on chromosome number 5. The child born with this is very severely disabled intellectually and usually ends up with dying early in childhood. There are also certain cancers that can be caused by translocations of chromosomes. Okay, so that ends my discussion of section 15.4. Please make sure that you read this section in your textbook. Don't forget the homework assignment that I assigned you yesterday on genetic linkage, and I will see you again tomorrow.